Okay, a couple quick announcements. While it is Wednesday, we do not have a Brightspace quiz today. So remember, no Brightspace quiz today. But that is just means a little extra time to prep up for the exam, which is on Monday. Remember, we don't have class on Friday. It's Good Friday break. But when we come back from Easter weekend, you guys will have a exam on Monday, the 18th in here. And that exam number 3 is going to cover the growth lecture set. So that's where we talked about things like the equations to describe growth, batch versus continuous culture. Talk about control of growth, enzymes and energetics, metabolism, and DNA replication and microbial genetics here. All of that stuff is going to be included then on the 3rd exam. Questions about that? Yeah.
For the equation questions, are they going to be multiple choice or is it going to be kind of short answer type?
Short answer. So they're short answer, so I'm giving you a little room to do the math and draw on the thing and write everything out. So you got the space to do that, but they are going to be short answer. Okay, so we're almost kind of at the end of the exam 3 content. So most of what we're going to talk about today is going to be material from, for instance, exam 4. So that's what we're going to get started here in a second. But I did think I would go back through PCR diagnostics. Then just to go back through that real quick. What is PCR? What is gel electrophoresis? Finish up that gel electrophoresis storyline then. So PCR, what does PCR do? Polymerase chain reaction. What's the overall function or purpose of a PCR? Just a PCR in general, not necessary for diagnosing disease. No, it's not here on the screen. Sorry, what? No, PCR is not going to be DNA to RNA involved. It is going to be DNA involved in the PCR, but something's going to happen to the DNA during a PCR, during a polymerase chain reaction.
It produces a lot of copies of like specific.
Yeah. It copies regions or segments of the DNA. Through a polymerase chain reaction, what that is doing is, if you guys ever think of a chain reaction, it is something growing, getting bigger, for instance. What isn't happening here with polymerase chain reaction is you're going from like a copy of the DNA molecule, but you're doing a copy of a specific segment, like one particular gene of interest. Remember, a gene is just a segment of DNA that codes for a final functional product. So that's our definition of a gene. So what you can do with a PCR is you can take one gene within all the DNA of an organism, but you're going to amplify up or make many copies of that one gene of interest. So PCR is going to amplify, replicate DNA, make many copies of the DNA, but just a specific segment of the DNA, not all of the DNA. What were some of the things you had to mix together in a PCR for it to work? How do you do PCR inside of a test tube? What were some of the components we had to put in? Primers. Yep, that was one of them. We designed the primers, and the primers are on either side of our region of interest. So we designed one primer on the left-hand side, we designed a primer on the right-hand side. What that means is our primers dictate what gene is going to get amplified, what segment of DNA is going to get amplified. So primers are really important. Template DNA, you need a DNA molecule to copy. Yeah, nucleotides then, and then the DNA polymerase, Taq DNA polymerase. So there's really 4 things you got to mix into their primers. You need a DNA that you can actually copy, so you need a template DNA. You need nucleotides to build new DNA strands, and you need the enzyme to build those new DNA strands, DNA polymerase. Taq DNA polymerase is more specifically what it's called usually. So, but you make many copies. So for instance, if I want to diagnose Mycobacterium tuberculosis, I create a primer on one side of the IS6-110 gene. This is a gene that is unique to tuberculosis bacteria. So if that gene is present, it means that tuberculosis bacteria is also present. So you put a primer on one side of that gene sequence, you put a primer on the other side of the gene sequence, and you use PCR to amplify that up. Again, if the patient has tuberculosis, that means they have that DNA segment in there, that DNA template in there. So if they have it, they get a DNA band replicated. But if they don't have tuberculosis, that means there's no template DNA present and their PCR is not going to work. Does that make sense, everybody? Or questions about that? So if a patient doesn't actually have tuberculosis, you won't get any PCR product at the end of it. You won't get any DNA amplified. But we use the primers dictate what is going to get amplified. We can't see the DNA, so that's where gel electrophoresis comes into play. We talked a little bit about gel electrophoresis last lecture as well, but you load your sample. You literally create a gel. So, and it's just like making Jell-O. It really is. Making gels is super easy in the lab. You just take a little bit of a powder called agarose, very similar to gelatin from Jell-O, just a little higher quality. And you just boil it. You got the gelatin, you got the agarose, you boil it, get it in a solution, you let it cool, you get a solid gel, for instance. Same way you make Jell-O, you boil the Jell-O, you let it cool, you get Jell-O, right? Okay. So what ends up happening though is when we pour the gel, we put a little, what's called gel comb in there that forms holes, wells in the gel, for instance. So those go at the top of the gel. And then we just load our DNA samples into those wells. All right, we actually just are going to pipette them in. What's called pipetting, pretty easy to do. And then we apply electrical field. We put a negative electrical field at the top, a positive electrical field at the bottom. Where something like DNA or RNA are electrically charged. DNA, RNA have a negative charge. So they are negatively charged. But what that means is, again, negative charges will repel other negative charges, and also negative charges are attracted to positive charges. So this is going to cause the DNA to what is called migrate, or just move through your gel. The thing is, the gel's thick, the gel's dense and viscous. So longer DNA molecules, again, the larger or longer the DNA molecule is, the more often it bumps into the gel matrix, and that slows it down, has friction. Okay. And then the shorter the DNA molecule, the further it moves. So longer DNA molecules, those are going to be closer to the well. Shorter DNA molecules are going to be more away from the wells where you originally loaded the DNA off. Questions about that then with gel electrophoresis? Okay, can't really see DNA, so you have to be able to visualize them in the gel when you're done. So you can't see DNA inside of that gel. There has to be a way to stain the DNA to make it easier to visualize and see. A lot of times DNA and RNA are stained with a compound called ethidium bromide. And ethidium bromide kind of does two things. It binds to DNA in general. That's one of the things it does. But it also fluoresces under UV light. So what's going to happen is that ethidium bromide, again, it will bind to DNA. So it tells you, binds to DNA, it locates the same spots DNA is as well. But it also fluoresces under UV light. So that's why you're kind of seeing this purplish, bluish glow here. What happened is you're seeing that gel under a UV light source. But that allows you to basically, because ethidium bromide glows under UV light, because it binds to DNA, it allows you to visualize your DNA bands. So that's what you're seeing in this particular image. You're seeing those DNA, where the DNA has run at the end of this gel electrophoresis then. Protein gels are, you can't really stain them with ethidium bromide. You need something else, but this is a protein gel right here. They use a special stain just called Comassi blue. But Comassi blue binds to proteins in general and has a really strong blue coloration to it. So it allows you to visualize where proteins are in the gel as well. Okay. The other important thing in all of this is, remember, gel electrophoresis separates out DNA, RNA, protein based upon the size of the molecules. So one other thing that's often put into these are what are called size standards or molecular ladders. So typically you can just purchase these, but a molecular ladder, this is for instance, a size standard or molecular ladder for a protein gel, but it is something you purchase from a biotech company. And it has proteins, a mixture of proteins of known sizes. So it might have a protein that has a mass of 14.4 kilodaltons. It has another protein in there with a mass of 20.1, 31, 43, 68, 92. What it does is it just tells you, hey, this protein here is, we know what size it is. And basically they're called size standards, molecular ladders, because you use them to interpret what is the size of your protein. Say you have an unknown protein on your gel. Well, now I know that this one's probably about 28 kilodaltons because I can come over and compare it against the ladder. Does that make sense, everybody? So you can size this. When you're doing disease diagnostics, for instance, your PCR product, your DNA molecule you amplify should be the right size. So in something like that ISX 110 example, you're looking for a 1,000 base pair long DNA molecule. And if you get done with the PCR and you get a 2,000 base pair DNA molecule instead, that's the wrong size. It's not what you're actually looking for. So the size standards are a little check to make sure your PCR worked the way it was supposed to work as well. So you just load these into a well, run them alongside your samples, but they ensure that you're getting the correctly sized PCR product you were anticipating. So it's a quick double check. Did the PCR work the way it was supposed to? Questions about that then? Or questions about PCR, gel electrophoresis, DNA replication? Also remember, PCR and DNA replication are very similar to one another. So technically speaking, if you're studying one, you're studying both. You just have to remember the key differences between them then as well. Okay, so you can't compare and contrast DNA replication versus PCR. Okay, questions then about exam 3. That's the end of exam 3 content. Any questions about that before I move on? How many of you guys have, well, probably most of you have had a PCR test done on you, unfortunately. Although they've moved a lot more towards antigen testing, for instance, for COVID-19, which is, we talked a little bit about that earlier as well in the semester, but a lot of times when they're doing those nasal swabs, the PCR tests are kind of the gold standard for testing. Usually PCR are accurate about 99% or more of the time. PCR diagnostic tests are the most accurate way to diagnose disease nowadays. So they're pretty important. That's why I wanted to spend some time going through what goes into those. All right, otherwise, if you guys do have any questions, stop me anytime. But we're going to start going through exam 4 content because we got this. The exam 4 is going to be a lot more about disease and immunity. So we're getting into that particular unit here. Genetic transfer is the first thing we're going to talk about as kind of part of this, kind of transition to this between microbial genetics and disease. But genetic transfer is going to come up a few different times as we talk about disease then as well. So first off, genetic transfer, there's different ways to transfer genetic sequences. By the way, what do I mean when I say genetic sequences? What's being transferred if we're talking about genes and genetics? DNA. Remember, genetics involves DNA. There's a few different ways that DNA is transferred. One is actually just reproduction. When I say that term reproduction, though, what do I mean by reproduction? What is reproduction? Everybody's like, oh, crud. That is often involved, that's more the central dogma of biology. That describes information flow within a single cell. So reproduction is going to be involving more than just a single cell. So yeah, central dogma is really important though. If somebody reproduces, what do? Offspring. So when we say reproduction, we're typically talking about some type of parent cell or parent organism. When they go through reproduction, they produce offspring then. So reproduction, there's a couple different ways to define that, but typically reproduction is passing on genes to a next generation. So if I had to kind of give you the most general definition of reproduction, again, it's passing on the genetics from one generation to a next generation. However, are there different kinds of reproduction? What are there's two main types of reproduction. What are the two main types of reproduction? Sexual reproduction. And then what's the other one? Asexual. So we'll come back to that in a second here. Sexual reproduction. If you guys haven't had a definition for this, this is where two parents combine genetic material into genetically varied offspring. Then we also have asexual reproduction. And asexual reproduction, does anybody want to hazard a guess on what asexual reproduction is? Maybe a definition for that one then? So are there two parents in asexual reproduction? No. This is when a parent reproduces genetically identical offspring. So the idea here, sexual reproduction is going to involve mating. Sexual reproduction, you're going to have two parents, they're going to combine their genetic material, but the offspring that are produced are unique genetically. They get genetics from both, say, the mother and the father, so they're not genetic copies of the parents. They're genetically varied compared to the parents. And a lot of times you'll also see they're genetically varied compared to siblings then as well. So if you have families, if you think about it, and you have multiple brothers and sisters in the family, they're not going to look genetically the same. They're not going to have the same traits. So sexual reproduction is going to be two parents combined and generating genetically varied offspring. Asexual reproduction is going to involve one parent producing offspring, but those offspring are typically going to be what is called clones. They're genetic identical copies of one another. So the offspring are identical to other offspring that are produced, but they're also genetically identical to the original parent then as well. So that's kind of the thing here. The difference between these is how many parents are involved, but also are the offspring genetically varied or are the offspring genetically identical? That's the two main things you're looking at the differences here. How do bacteria reproduce? Sexually or asexually? Yeah, they're going to do asexual reproduction. Again, something like binary fission is a type of asexual reproduction, where then you have one parent cell copies its DNA. Typically, that DNA is copied during DNA replication, and they're identical. Occasionally, you get a mutation or three. But generally speaking, the chromosomes are identical down to the base pair, copies of one another, and then you get identical daughter cells produced. Questions about that? So again, binary fission, if I had to give you a definition of that, a parent cell gives rise to two identical daughter cells, but again, identical genetically, identical in size then as well. This reproduction is sometimes referred to as vertical gene transfer. Vertical gene transfer is where all of the chromosomes, all of the DNA is then going from one generation to the next via reproduction. So vertical gene transfer is about reproduction. And this is because you're going from one generation down into a next generation, reproduction. And again, sexual reproduction or asexual reproduction is collectively sometimes also referred to as vertical gene transfer. Now, and this is, again, bacteria are going to do vertical gene transfer through some process like binary fission. Okay, one generation to a next generation. That's vertical gene transfer. That being said, again, bacteria, they don't reproduce sexually. Again, we just said that. So bacteria do not reproduce sexually. They reproduce asexually. That doesn't mean they don't get a little promiscuous sometimes, though. So bacteria do sometimes, they don't do sexual reproduction, but they like to grab DNA and try it out. So what ends up happening, bacteria can often exchange small gene segments, and they tend to do so pretty frequently. So they might grab not all of the DNA, but they might grab a gene. And when they pick up a gene segment or a few gene segments, this is then referred to as what is called horizontal gene transfer. And I'm going to show you a diagram that helps make this more sense here in a second. But this is transfer of one or a few genes from one existing bacterial cell to another existing bacterial cell. No reproduction involved. You already have two cells. They're already there. And you get some genetics, some genes moving from one, the donor cell, into another, the recipient cell. And so it's called horizontal gene transfer because there's no reproduction involved, and it's horizontally transferring from one existing cell into another. And again, it just looks like this. This horizontal gene transfer just looks like this. You got a donor cell, it's got some DNA inside of it, and not all of the DNA moves from the donor to a recipient cell. But some of the genetic sequences, a gene here, a gene there, is going to move over from a donor cell into a recipient cell. So this horizontal gene transfer does require two already existing cells in some way. It does not involve reproduction. And bacteria do this all the time. And they pick up a gene here. They pick up a gene there. But when they pick up genes, they pick up what that gene encodes. And when they pick up that functional product, say an enzyme, they can gain new traits. They can gain new abilities, in essence. And this is really a big deal. You think, oh, it picked up a gene here and there, but actually picking up a gene here and there can actually be really important. So for instance, there's E. coli K12, we talked about towards the beginning of the semester, versus E. coli 0157H7. K12 is a bacteria that is in your gut and it's good for you. It's a mutualistic species. 0157H7 is actually a pathogen and causes food poisoning. And the difference between them is about 10 genes. About 10 genes to convert something that's good for us into something that makes us sick. And how do those genes get in there? Gene transfer. So we're going to talk a little bit about this when it comes related to some disease that we're going to talk about. How do bacteria become antibiotic resistant? Gene transfer. And we're going to talk a little bit about that when we talk about antibiotics and antibiotic resistance here in a few lectures then as well. Okay. So when it comes to gene transfer, there's three main different mechanisms by which bacteria can do horizontal gene transfer. Transformation, conjugation, transduction. These are all ways that horizontal gene transfer happens, but they're different mechanisms driving that horizontal gene transfer. So we're going to talk about each one of these three At a time, we're going to talk about transformation first. This is uptake of DNA free floating out in the environment into a recipient cell. So transformation, again, this is just uptake of DNA that's kind of free floating in the environment. But how do you get DNA that's free floating in the environment? Well, you might have a donor cell. So this is my donor cell. It's got its chromosome, its DNA is this green circle. Remember, bacteria have circular chromosomes. What ends up happening though is it dies. So my donor cell dies, it breaks open, undergoes lysis, breaks open. The DNA often gets kind of destroyed. It breaks down into short fragments. And maybe each one of those fragments is carrying 1, 10, 15 genes. So the DNA fragments apart. But now these DNA fragments carrying just a few genes are free floating out in the environment. Well, some little recipient cell comes along, and if it's capable of transformation, then it can take up that DNA, and sometimes it can incorporate that DNA into its chromosome. So it picks up the genes from this DNA fragment and then starts to use those genes for itself then as well. So other cells, the recipient cells, come up and take up that free DNA. Now, some species of bacteria can do this and some species cannot do this. So for instance, just to give you a couple examples from the ones you've been working in the lab, Escherichia coli can't do transformation. If you just take some DNA, put E. coli in a DNA bath, for instance, nothing's going to happen. But another species you guys work with in lab is Bacillus subtilis. It can do transformation. Bacillus subtilis can take up DNA from its surroundings and incorporate those DNA fragments. And it's basically, do they have something called competence machinery enzymes or not? Competence machinery. This is a DNA, is a series of enzymes that act as a DNA import system. Some bacteria have these enzyme complexes, like Bacillus subtilis. Some bacteria don't have these, like Escherichia coli. So something like Bacillus subtilis, just because it has the COM system, these enzymes right here, it can do transformation because it has these enzymes. So the COM machinery is actually probably a modified pilus structure. Don't worry about that too much, but they allow DNA import. And so bacteria that have COM machinery can do transformation. Bacteria that lack COM machinery can't do transformation. And really what it has is if they have these COM machinery, it's a DNA import system. So it's a series of membrane embedded proteins. First, they bind to double-stranded DNA. They actually digest that double-stranded DNA down to one strand. So they actually break apart one of the strands of DNA and they keep the other strand. That single-stranded DNA is then taken up and internalized into the cell through the competence system, through the COM system. And then it can be recombined and put into the bacterial chromosome. So it actually will integrate into the bacterial chromosome and become part of the bacterial chromosome. And in essence, then the bacteria has picked up new genes it did not have previously. Questions about that so far? Now, why do this? Why do they want to pick up genes? Well, there's a few different reasons. Again, think about this though. The DNA fragments were free floating. That means the donor cell died, broke open, and then it's got DNA from a dead cell. So why would a bacteria want to take up DNA from a dead cell? That dead donor cell wasn't very successful. It died. But this doesn't mean all of its genetic sequences are bad. Yeah, maybe it died because it had a bad gene or two. Maybe it had a bad version of a gene. Maybe some of its genetics was actually good still. So what's going on here? Why do competence? Why do transformation? Maybe this is a way for bacteria with the recipient cells to kind of sample their genetic environment and find maybe a lucky penny down on the ground, but instead they're trying to find good genes. Maybe they'll get lucky and they'll pick up something that's good for them, a beneficial gene, and whatever that gene codes for. Maybe this actually evolved as a way of eating DNA. I actually said these competent systems, they bind to DNA, they digest it. They actually can use those digested nucleotides as a source of nucleotides to build their own nucleic acid. So this may have actually originally evolved as a way of eating DNA as well. It might also be that it's repair of damaged gene sequences. Well, maybe this donor cell died because of, say, radiation, and some of its genes got mutated, destroyed. But maybe other parts of the donor chromosome were fine. Well, maybe this recipient cell is also getting exposed to radiation, and some of its DNA is getting damaged too. But maybe gene 2 is getting damaged here, whereas gene 2 was intact over here. Maybe here is gene number 10 got damaged. That killed the cell. But this guy can pick up a copy, a good copy of gene 2, and repair its bad copy of gene 2. So for instance, sometimes this is a way of repairing also damaged gene sequences. What this really is, the real reason isn't known. It's probably actually a combination of all three of these, just depending upon the surrounding conditions. Then probably all three of these are valid reasons for why to do competence or transformation. Questions about that then?
Can it pick up bad genes like the recipient? If it's like
Yes, the recipient cell can pick up bad genes, and sometimes that's bad for the recipient cell. It is very much almost kind of like buying a lottery ticket. They're hoping to get lucky. All right, that leads me to a pathogen of interest for you, Streptococcus pneumoniae. This is a gram-positive bacterium. It grows in coccus arrangements, and it causes pneumonia. So it affects the lung. And again, here you can kind of see, this is a chest x-ray, but all this stuff is not supposed to be occluded. It's supposed to be clear. And this is actually the bacteria growing up enough in the lung that it blocks the x-rays. And this used to be a very common way to diagnose pneumonia cases, for instance. They even did it early on in the COVID-19 pandemic. One of the early on, before they had PCR tests, one of the best ways to diagnose COVID-19 infections was also chest x-rays, because it would show damage to the lungs, for instance. So causes pneumonia. Streptococcus pneumoniae can also sometimes cause ear or sinus infections. And in rare cases, it can cause bacterial meningitis. So this is going to be when bacteria get into the fluids of the spinal cord and brain. But this causes inflammation of the spinal cord and brain that can be fatal, for instance. So bacterial meningitis is usually pretty serious. Yeah, it can also sometimes even cause blood infections, which are then referred to as septicemias or bacteremias, then, for instance. It also turns out most people do not die directly from the flu. You might have heard like, oh, there was this many flu deaths per year. So for instance, in 2013, which was one of the higher flu seasons, it's estimated about 65,000 people died in 2013 from the flu in the US from that particular flu season. That was one of our more severe flu seasons in the last like 2 decades. So the problem is most people don't die directly from the flu. The flu makes them sick. But because they're sick, they become immunocompromised. They become a compromised host. Something we're going to talk more about here later. But that makes them vulnerable to a secondary infection that they normally would not get otherwise. So what most people die of from the flu is they die from a combination of flu and pneumonia. So they get the flu, that makes them vulnerable to pneumonia. They get pneumonia, and then it's that combo that typically kills people. That's in contrast to COVID-19, which just kills people. All right, that's why COVID-19, one of the reasons why COVID-19 is considered much more severe than the flu. So most deaths from the flu virus actually result because of a secondary infection from Streptococcus pneumoniae, causing also a flu and then pneumonia case after that. Transmission of Streptococcus pneumoniae is primarily coughing, sneezing, close contact droplet transmission. We're going to talk about more some of that types of stuff. And it does cause about 175,000 hospitalizations per year in the US. So when you guys are working with nurses in the future, you're going to see a lot of pneumonia patients, and this is probably what they have. Remember, walking pneumonia, we talked about that earlier in the semester, is caused by Mycoplasma pneumoniae. Regular old pneumonia, which is usually more severe than as well, is then caused by Streptococcus pneumoniae most commonly. Although there's a couple other bacteria that can cause it as well. Typically then treatment is antibiotics, and they also then have vaccines to prevent these. So they have a pneumococcal vaccines, and it is really recommended that anyone over the age of, I think about 55 get that particular vaccine, because usually the older you are, the more vulnerable you are to pneumonia cases.
Was someone with, Streptococcus pneumonia, would they be on droplet precautions in the hospital?
They can be, especially if they're not responding to antibiotic treatment. So there are some antibiotic resistant varieties of Streptococcus pneumonia, and that particular patient would definitely be on droplet treatment then. Yeah. If it's an antibiotic resistant strain not responding to antibiotic treatment, they do not want that spreading out to more people. Why am I bringing up Streptococcus pneumonia here? It's actually capable of transformation. So it has the COM machinery enzymes, so it can do transformation. It can take up DNA from its surroundings. It's also pretty interesting in that it has a lot of what's called antigenic variation. We're going to talk about antibodies and antigens a little bit here soon. But it turns out Streptococcus pneumoniae is capsulated. Do you guys remember what a capsule is? It's been a while. What's a capsule?
It's like an extra layer that like replication and other extreme.
Yeah, so it's an extra little layer around the cell, but what's that layer made out of? Sugars, extracellular polysaccharide layers. So the capsule layers are a layer of sugars that wraps around the cell. But it turns out Streptococcus pneumoniae makes 90 different versions of a capsule layer. It can actually make capsules out of 90 different sugars. And sometimes it makes one sugar and then it switches over to a different one. And then it can switch over to a different one. And then it can switch over to a different one over time. And what this does is by having different capsule layers, every time it switches from one type of sugar in its capsules to another, it looks different to our immune system. So it has this variation. It makes many different capsules to evade our adaptive immune system. Every time it changes the capsules, it's like it's a brand new thing the immune system has not seen before. So that they have trouble responding to it. The other thing is some of these capsules do what is called molecular mimicry. Some of these capsules look like sugars, normal human sugars. So some of their capsules, like say this one, actually looks like normal sugars that our cells also make. And it tricks our immune system to say, this is fine. This is not a problem. Nothing wrong here. And so our immune system doesn't recognize it because it looks like host cell sugars. Questions about that? That capsule is required for virulence. Virulence is the ability to cause disease. So there's a pretty famous set of experiments. And yes, if you had Bio 1, you already heard about these experiments a little bit. But I'm not going to go back through it in that level of detail. But there's a pretty famous set of experiments by Avery and his research group in 1930. And what they did is they had a version of Streptococcus pneumoniae that was defective for capsule production. And if they infected the capsule-less strain, no capsule, into, say, their host organisms, my mice, no disease. The version of Streptococcus pneumoniae that doesn't have a capsule is actually harmless to animals and does not cause disease. But if you have the variety of Streptococcus pneumoniae that then has a capsule layer that gets in, it helps protect it from the host immune system. And that means it can grow up in the host and cause damage to the host. And so with the capsule, Streptococcus pneumoniae is virulent, disease causing, and my little mouse died. So it turns out capsule is essential for virulence. And by the way, a lot of disease causing bacteria are capsulated. Most of them are actually. Most of them have EPS layers, extracellular polysaccharide layers. Now, what this experiment also showed was genetic transfer though. So the other thing they did is they took the capsule strain of Streptococcus pneumoniae, but they killed it with heat. So dead bacteria. And what happens is when they just put only dead bacteria into the mouse, nice, healthy, no problem. But what they showed is that if you took the heat-killed capsule strain, so it's dead, and then you actually mixed it together with normal living capsulus strain, what ends up happening is you would think this guy's harmless. You put it in the mouse, you think the mouse will be fine. No, the mouse died. What ended up happening is gene transfer. The capsule genes, the genes responsible for making capsules, were made their way into the harmless strain. And so picked up those genes by transformation. It gained the ability to make capsules and then caused disease. And in fact, when you took dead capsule strains mixed together with live capsulus strains, what you get out of the mouse is capsule making strains. And this is because again, the capsulus strain in green picked up the capsule synthesis genes, became capable of making capsules and capable of then causing disease. This was the very first evidence for transformation or horizontal gene transfer in bacteria. Questions about that? All right, that's transformation. What about the another type of then horizontal gene transfer? Conjugation. Conjugation, this is the export of DNA from one cell into a recipient cell. And it requires an enzyme complex called the TRA machinery. It also requires something called an F factor. So conjugation involves an F factor, and an F factor is actually just a specific kind of plasmid. We talked about plasmids earlier on in the semester when we talked about structural anatomy of a bacterial cell. A plasmid is a small circular piece of DNA with its own origin of replication. All right, so a bacterial chromosome is usually about, on average, say 5 million base pairs long, 5 million nucleotides long. A plasmid is then typically about 5,000 to 20,000 base pairs long, so much, much smaller. Okay, chromosome's big, plasmid's a little tiny. But that plasmid still carries genes. And so and the F factor is a specific version of a plasmid. It's a plasmid that is carrying genes that codes for something called a sex pilus. It carries genes that codes for the TRA machinery, which are involved in the DNA transfer. And it's a plasmid that has its own origin of replication. So that means that because it has an origin of replication, anytime the cell goes through DNA replication, it's going to make a copy of the chromosome, but also it's going to make a copy of the plasmid as well. So the origin of replication means DNA replication is going to copy that as well. So what happens during conjugation is you have this F factor plasmid. Again, this might be a bacterial chromosome. This is the F factor plasmid up here. What's going to happen is you have a donor cell. My donor cell is in the brown here. The F positive means it's got the F factor and it's going to be my donor cell. This is where the DNA is going to move out of. It's going to donate the DNA to another cell. Then you have an F minus. Maybe this is a recipient cell. It doesn't have the F factor. What's going to happen is a pilus is going to extend out from the donor cell and it's going to attach onto the recipient cell. And it then retracts. It kind of reels it in. And as it retracts, those two cells get into very close proximity to one another. In essence, they dock together. So the pilus retracts and it brings the two cells into contact. This is then going to allow the TRA machinery to then work. The TRA machinery is what's going to accomplish the DNA transfer. The plasmid is going to get replicated by DNA replication. A copy gets made. And then that copy of the plasmid is transferred through the TRA machinery from the donor cell into the recipient cell. So we're just showing that up here in my little diagram then. And the recipient cell now gains a copy of the F factor plasmid. But remember, it's a copy. So the original donor cell is still there, still humming along, still has a copy of the F factor plasmid. But now the recipient cell got a copy of that and is also now F positive in and of itself. And that can go on and spread those DNA sequences to more cells. So the recipient in this cross is now also can be act as a donor cell to additional bacteria over time. Questions about that? Here's the thing about all of this. The plasmid, to be an F factor plasmid, it has to have these. But it can carry more than that. Sometimes on these plasmids, there's also things like virulence genes, genes involved in causing disease. Sometimes there's also on these plasmids antibiotic resistance genes, genes that make a bacteria resistant to antibiotic treatment. So sometimes there's some, these are the genes that have to be on there to be considered an F factor plasmid, but sometimes it can carry additional stuff and transfer genes between bacterial cells then as well. Questions about that? All right, then I actually have another pathogen of interest for you. That's probably what we'll end on for today. But pathogen of interest for you, Agrobacterium tumefaciens. This is not it is a pathogen, but not of humans. It's actually a plant pathogen. So I'm mixing it up on you guys for once. All right, so this is a plant pathogen. It actually creates what are called gall tumors. So this is, for instance, a tumor on the plant, but it's often referred to as a crown gall. So if you've ever seen a plant with like this, something like this, it's probably infected with Agrobacterium tumefaciens then. Agrobacterium, this is maybe my Agrobacterium. It turns out it has a type of plasmid called a TI plasmid. And that TI plasmid can get transferred into plants by conjugation. So in this case, it's the bacterial cell will use conjugation to transfer genetic sequences, the TI plasmid, not into other bacterial cells, but into a plant cell. And my little green rectangle here is supposed to be my plant cell. Okay. When that TI plasmid gets into a plant cell, it introduces genes into the plant cell. But specifically, it introduces a growth hormone gene. It causes the plant cells to make high levels of growth hormone. And when a plant cell is making high levels of growth hormone, it divides. And it makes those crown gall tumors. If you guys think of what a tumor is, it's basically a mass of cells rapidly undergoing unchecked cell division. So when we talk about cancers as tumors, what a cancer is, it's a bunch of cells that just keep dividing, and they make a mass or a tumor. In this case, the bacterium is literally causing plant cancer. It's making these tumors where the plant cells just keep dividing and unchecked. It also then causes those plant cells to start making octopines. And this is actually an example of an octopine molecule, but it's a carbon-rich, nitrogen-rich, energy-rich molecule. It's a food source then for the Agrobacterium. Okay, with this, the way it works, again, this might be my little tiny Agrobacterium cell. This is my bigger plant cell because it's eukaryotic. Agrobacterium transfers the TI plasmid into the plant cell by conjugation. It makes its way to the DNA of the plant cell and it integrates into the plant cell DNA. So the TI plasmid then gets recombined into the plant cell chromosomes. So that's what you're seeing here in this image. TI plasmid is this guy. Now it's in my little chromosome. This is supposed to be the nucleus of the plant cell, and the chromosomes, the linear chromosomes of the plant cell, it integrates into it. Causes that plant cell to start making growth hormones. And because it's making growth hormones, the plant cell divides over and over. So you're seeing my plant cells multiply and all of them are infected. It also causes all those plant cells to start making octopines. And this then acts, again, it's a carbon, energy, nitrogen-rich molecule that only Agrobacterium eats. And the Agrobacterium feeds on those octopines and then they increase in number so you get more of them. So that's kind of what's happening here with Agrobacterium. Now, Agrobacterium, again, plant pathogen. Last thing I wanted to say, and that's what we'll finish up here for today. It's also heavily used to genetically engineer plants. Turns out you can use that TI plasmid. You just take out the growth hormone gene. You take out the octopine synthesis gene. You replace them with whatever gene you want to insert into plants instead. So maybe you want to make a herbicide resistance gene. So you take the TI plasmid, you modify it, you put an herbicide resistance gene into it, and then you have herbicide resistant crops. So you have corn, wheat that you can spray with herbicides to kill the weeds, but doesn't affect the crops that the farmer is growing. And yes, that is done all the time by some of the agricultural companies. For instance, you probably have all seen like the hybrid corn signs that all the farmers are putting up in their fields and all the stuff like that, but those are genetically modified crops, all of them. Questions about that? Okay. All right, that's what we'll stop for today. We'll pick back up with transduction on Wednesday after the exam. What did I modify? Cash. Never has. Yeah.
Okay, that's
Yeah. No, it will not match up. I teach in the micro is my expertise. So the textbook's really good. That's a really approachable textbook, but you do kind of have to pay attention to what I posted I'm going to cover, and then read the corresponding sections and maybe ignore the
So
others.
like, on these, you have like.
Is it not matching up there?
No, like I tried looking through chapter 6.
It should be. Oh no.
Yeah.
Is it for genetics? Says I
So, like,
must
I
have
was-
I must have missed copy. I modified the schedule. I bet I missed copying and pasting some stuff around.
Okay. That's
I'll take a
fine.
look.
Okay, just wondering.
Yeah, sorry about that.